Hello, everybody. This is going to be a quick review of how mutations to the different genes in the Lac Operon affect gene expression. And hopefully this will help you with your homework. So let's get into it. The Lac Operon is usually repressed, which means that it is an inducible operon. I will go more into this in the upcoming review video, but that basically means that some protein will attach to the repressor, causing it to pop off the operator, so transcription can happen, and it is induced. It's usually off. That's what it means by when we say it's usually repressed. These are the elements of the lac operon. This picture has most of them, a few that are shown in different things, so I'll explain. We have lac I. This is its own little gene that codes the repressor protein that binds to the operator. So this has its own promoter and own regulation of transcription and translation. So if this is functional, meaning it's lac I plus, then the repressor protein will be made as usual and it will bind to the operator until um, the correct protein binds to the repressor protein, and it leaves, allowing transcription. So this lac I is responsible for regulation of the gene. We also have, this is shown as lac P. It's the promoter, where RNA polymerase binds. And then we have lac O, which is the operator. If either of these, the normal type is lac P plus and lac O plus. Changes to these, like say if it's lac P minus, that means, or lac O minus, that means that the promoter or the operator are inactive, RNA polymerase cannot bind, or it changes how the repressor binds, and so this will affect transcription because if RNA polymerase can't bind, then the gene can't be transcribed, or if the repressor protein can't bind, then this won't have normal regulation because this will always be transcribed. Um, or if the repressor protein is always bound, then this will never be transcribed. So that's kind of the deal with lac P and lac O. We also have lac Z and lac Y. So lac Z, lac Y. We also have lac A. We don't actually really know what lac A does, and so therefore it's not important for the purposes of what you should know about the lac operon. So we have lac-Z, which creates the protein that actually cleaves the lactose and breaks down the lactose for you. And then we have lac-Y, which helps lac-Z do this. So without lac-Y, without a functioning lac-Y protein, which would be lac-Y plus, lac-Z cannot function. Likewise, even if you have lac-Y plus, but you have lac-Z minus, meaning non-functional lac-Z, then the proper lac-Z is never produced, and therefore the protein, it won't be able to cleave lactose like usual. So both lac-Z and lac-Y need to be positive for the correct protein to be produced and it to have the right function. So what happens when we have multiple strains? Also, I have a breakdown of this. If you would like to screenshot it, look at it for a second, you can pause the video and do so. Um, so what if we have two different strains? Let's say we have lac I plus, lac P plus, lac O plus, lac, um, lac Y negative, and lac Z plus. So what would happen in this strain? Well, lac I is plus, so that means the correct repressor protein would be made, meaning this would have normal regulation. Additionally, the promoter is active and the operator is active. These are all regulatory elements and determine if transcription or translation happen or not. Because all of these are positive, it does happen. So this, we have a check mark. All of the transcription is working properly. We then have lac Y and lac Z. So lac Z is positive, meaning yay, that one's good. but lac Y is negative. And since lac Z relies on lac Y, it will not be able to break down lactose because lac Y is not produced. Okay, let's look at another example. What if we have lac I minus, lac P plus, lac O plus, lac Z, no, let's do Y first. Lac Y plus lac Z minus. Okay, so in this one, we have lac I minus. That means the repressor protein will not be produced. This means that this will continuously be transcribed. So already we don't have normal regulation, but these will still be produced, meaning we have an opportunity to break down lactose. Or do we? Because if you see here, our lac Z is negative, which means that protein is non-functional, meaning this will not break down lactose. So even if lac I was plus, because lac Z is negative, it will not break down lactose. We can think of these in general as a huge chain of events. So they all rely on one another to keep the train moving, to keep normal functioning gene expression. Now we can get to in-trans regulation. So that's, let's say, a bacteria has one operon that comes with part of its genome. This is lac I minus, lac P plus, lac O plus, lac C plus, lac Y plus. So this is the one that comes with the genome. And then we can also sometimes have these things called F plasmids. I will go into this more in the review video, but basically these plasmids contain mutated versions of these genes. So this F plasmid contains another lac operon, and this is lac I plus, lac P minus, lac O plus, lac Z minus, lac Y plus. Okay, so let's look at these two. Each of these don't have normal regulation by themselves. We have lac I minus, uh, means the repressor protein will not produce anything, meaning that this will be transcribed all the time. But that's not great because then we're wasting useless resources, and this is getting transcribed all the time when it's supposed to be off most of the time. On this strand, the promoter is inactive, lac Z is inactive, like this cannot work on its own. However, it has the lac I plus, and this, since it's running on its own promoter and transcription, is still producing regulatory proteins. And these regulatory proteins can float over to this operon, where this operon is being transcribed and translated, and bind to the operator to make this have normal regulation. When these two interact like this, where the proteins from one can float over to the other and regulate it normally, or um, combine to help function, we call this in-trans regulation. So they are regulating between themselves. They're not regulating in-house. They're kind of outsourcing the different proteins. This is actually very useful for a lot of organisms because if they get a mutation, if they have another like F plasmid in prokaryotes, then it's not the end of the world. They can still function normally. So that's a bit of a review on the Lac Operon. I hope that was helpful for you, and I will see you in the next video.